right, ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here. ETM Hotel, Rennie Shawn, welcome in peace. My name is Shawn. I'd like to thank everybody for tuning in to Science with Shawn this evening. Appreciate everybody that is already in the building. Like the show, share the show, tell a friend that Shawn is back at it again. We're going to deal with some science. We're going to get straight to it. I know it's late. And some of y'all might rock out with me, others may not. But again, I need you to like the show, share the show, like the show, share the show. We got some news from the FDA today. For those who did not have the link, all the sources are in the description. Please support the merch store and the bookstore. Please, we have that. We have all of that right there for you in the description of this particular video. Man, hold on one second before we get going. I want to make sure that I get the video out to the correct people and the link. Yeah, it's Science with Shawn. We're getting it in. All right, so before we get into basically everything, I want to start the show out talking about the latest news with the FDA approval. We already went over this 2 days ago, and we've been going over it the last. We went over yesterday, and we in a little bit more, not really much more detail than we did the day before, but the science is out. And we're seeing that Moderna moved swiftly to get an EAU so they can start administering. And I believe next week we will start seeing Moderna being distributed across the country, which would also raise up the amount of the vaccine that's available. So we know really Pfizer has 10 million people covered, where there's going to be a refill on that, and they're going to make sure that they get those people covered. And now Moderna is going to be in action, and they're going to be able to cover a few million people, and we're going to be able to see more results and things of that nature. AstraZeneca hasn't chipped in yet, so we should be getting phase 3 trials from AstraZeneca. That would also help facilitate more data, and we kind of deal with it from there. I was watching CNN earlier, and I was listening to a doctor on CNN explain Bell's palsy, right? And how they were looking at things. But more importantly, I was listening to them just kind of get through everything. And as far as the fear of a future pandemic, and the doctor said, we've been dealing with mRNA for 25 years. He said, now with all the technology that we have, we're going to be able to make vaccines faster than we did just this previous time. That's what the doctor said. So I thought that was dope. To know, and I wanted to share that. Hold on, I'm still getting the show out. I don't expect a lot of people to show up, but yeah, like the show, share the show, support the merch store. I mean, it is the holidays. We've been working hard, put together merchandise for people to support, and COP this winter, this summer, this spring, this summer, this and next fall. I mean, come on, man, support us. Support us. All right, so let's deal with the FDA advisory panel endorses Moderna's COVID-19 vaccine. This article is in the description as well. I don't want to linger too much on it. We already know the information behind it. It says an advisory panel on Thursday recommended that the FDA administration issue an EAU or emergency use authorization for COVID-19 vaccine developed by Moderna. All but generating the agency will do so. Now, authorization expected on Friday will mark the second COVID-19 vaccine cleared by the agency and amount to one more step toward curbing a pandemic that has infected and estimated 17 million people and killed more than 300,000 in the United States. Both Moderna vaccine and the one developed by Pfizer and its partner BioNTech, which was granted an EAU last week, use messenger RNA to instruct the body cell to produce copies of the spike protein found on the exterior of the SARS-CoV-2, the virus that causes COVID-19. Those vaccine-induced proteins teach the immune system to recognize the coronavirus as an invader and attack it when an immunized person is exposed to the virus. All right. So clinical trials show both vaccines were highly effective, about 94 to 95%. In fact, though, both trigger side effects in a portion of people vaccinated. We know State Live blogged the meeting of advisory panel known as the Vaccine-Related Biological Product Advisory Committee is archived above. I'm not going to get into the resounding vote and a single doubter. It was overwhelmingly like 16 out of the 17 or how many other people voted. My bad. There you go. 20 votes in favor. One was delayed. One person was delayed. So out of all the people, the overall panel was rocking with it. Now, that information is in the thing. I want to deal with that. We've already dealt with the adverse effects. There have been mild adverse effects. We know how many people got what, when, where, and how. We went through this already, but if you don't think we got the documents, for anyone new listening in the audience, here's your FDA briefing. We had it 2 days ago, and this is what was published and handed out and read over. The draft was read over and convened over. Now I heard from Dr. Hildreth again today on CNN. I'm pretty sure that they're going to have him on C-SPAN again. When they do, I'm going to make sure that we review his work on Science with Shawn, and we'll deal with that. Also, another thing, Vaccines and Related Biological Product Advisory Committee December 17th meeting announcement. I put this in the chat. This is the actual YouTube stream, or you can click on the stream here to view the entire webcast. It's 8 hours, and it covers the whole entire meeting. So if you got 8 hours of your day to sit there and you want to listen to the entire FDA approval, feel free. You might learn some stuff. All right. So, every last article we have up here is there. And that's that on that. Let me look in the chat. See who we got in the building before class start. Peace, Brother Chavis, Queen Cheetah, Anusha, Benita. Peace to everybody. Peace to all the people who may catch the stream later. Shout out to y'all. Man, I appreciate y'all showing up. I know it's nighttime and y'all could be doing anything else. But let me go ahead and let's get this thing set up. I'm trying to delay it a little bit. There are some people that are not saying anything. There's a couple of viewers that are not saying anything. So I appreciate y'all tuned in too. And everybody else that's hopping on right now. But we'll hop on. We're going to go ahead and fire up this class. We're going to go right over here to the science behind the historic mRNA vaccine. This was published today at 4 a.m. this morning. And you see the sister on the front of the cover being administered by a sister. There you go. Yeah, this might be a political move. So what? It was a sister behind the creation of the vaccine for Pfizer. So there you go. You got a sister that was, front line in the vaccine. You have a sister who the first one to get it, and you have a sister that's the first one to administer it. All of that is historic. Creator, administer, and receiver. Period, point blank. You have women breaking ground. All right. So instead of reading over this, this article in this small length, and what we're going to do is we're going to listen. We're going to listen in on the article, and then we will get into our mRNA.
This message comes from NPR sponsor Scholastic, presenting Cat Kid Comic Club, a new graphic novel series from the creator of Dogman. In a starred review, Publishers Weekly called Cat Kid Comic Club irreverent and laugh out loud funny. Get your
All right. So I know y'all heard that, but I'm going to share my screen because I wasn't sharing nothing earlier. And I'm going to run through it again just for everybody. There go your sources. All your sources for anything that I read, right, are in the description. Everything is in the description. All of this. That's Moderna stuff that we had 2 days ago. You already seen this in the description. Here we go with the science behind the historic mRNA vaccine. There you go. The two sisters, the administrator and the receiver, and the creator is somewhere looking in.
copy today. Jeff Brumfield. What's up, Jeff?
Hey there.
Hey now.
Hello.
Order Ping Huang. Hiya Ping.
Hi, Maddie.
In a while.
Hi, Maddie. I miss you so much.
I know, I miss your face. If you've listened to Shortwave even a few times, you know our show is only possible because we're backed by so many reporters across NPR. NPR East Africa correspondent Ader Peralta. Hey there, Ader.
Hey, Maddie.
Lauren, welcome.
Hey, thanks for having me.
Rebecca Hersher. Hey, Becky.
Hey.
So, not to mention the producers, editors, and engineers that you hear in the credits pretty much every day.
Plus, the big one, you.
You are an amazing listener.
See, my gig, Emily's gig, everyone we just mentioned's gig relies on listener donations to your local member station. By supporting them, you're supporting us.
Exactly. We're in this together. It's because you find it in your heart to donate that we can bring you the latest news on the coronavirus.
Plus, all that science joy you can handle.
So please go to donate.npr.org/short for another year of Shortwave.
Yes, for the love of science itself. That's donate.npr.org/short. You're listening to Shortwave from NPR. Maddie Safai here with Emily Kwong.
Hi, Maddie.
Hey, you. So recently, we've had some sobering coronavirus news. Not only have we passed 300,000 coronavirus deaths in the US, but for the last 2 weeks, COVID-19 was the leading cause of death, ahead of heart disease and cancer.
But that news was contrasted by something hopeful on the vaccine side of things. News that began with a simple meeting.
And I will read the votes aloud for the record. Dr. Moore voted yes.
Following the vote of a panel of independent medical experts.
Dr. Carilla voted no. Dr. Rubin voted yes.
On Friday, the Food and Drug Administration moved to authorize the emergency use of the Pfizer-BioNTech coronavirus vaccine.
It's the first COVID-19 vaccine to be authorized by the FDA. It's also the first authorized vaccine to use something called mRNA technology. And to be clear, this is not an approval by the FDA.
Right. Emergency use authorizations are faster and have fewer steps.
I mean, it is an emergency after all.
But Pfizer-BioNTech still went through rigorous safety and efficacy trials in order to get this authorization. And the CDC also gave its endorsement the day after for use by people 16 and older in the US.
And Monday, Sandra Lindsay, a critical care nurse at Long Island Jewish Medical Center, became the first person in the US to receive the vaccine.
I hope this marks the beginning to the end of a very painful time in our history.
This week, many other healthcare workers are following suit, taking the first of the 2-dose vaccine.
Which is why today we're talking about the science behind the vaccine, how it works in your body, and how it was developed so quickly. You're listening to Shortwave from NPR.
Okay, Maddie, so today we're talking about how mRNA vaccines actually work. Where should we start?
So, in order to get more context on this vaccine, I called up Rene Najera. He's the editor of the History of Vaccines project with the College of Physicians Philadelphia. Rene is also an associate of epidemiology at Johns Hopkins Bloomberg School of Public Health. On top of that, he is a senior epidemiologist in a health department in the DC area. Plus, he's got a little one at home.
I gotta warn you though, the toddler is on her way home, so they'll be here like in 45 minutes, and she is loud.
That's really, really cute.
Yeah, he says, even though we've never had an mRNA vaccine licensed in the United States before, the basis for this technology has actually been around for a while.
Yeah, the 90s and the turn of the century is when the actual hardware to do this, the actual techniques in the lab become more well understood. It's only now that you have this true global pandemic, global emergency that pushed that technology to mature rather quickly.
And like we've talked about on the show before, this speeding along was the result of a huge effort by the global scientific community. Pretty much as soon as scientists got their hands on the genetic sequence of the virus in January, they were off to the races developing a vaccine.
Right.
And like we said earlier, this is an emergency use authorization, which by its nature happens pretty quickly.
Yeah, and another reason this vaccine came along quickly is because it's just simply quicker to make in the lab compared to like the flu vaccine or other viral vaccines.
Yeah, those are the vaccines that I'm honestly more familiar with. So let's start there. How do they generally work?
So they all work by giving your body a little taste of a potential virus, just enough for your immune system to recognize, hey, there's something in my body that is not mine, react to that intruder, and build up immunity to that virus.
Right. So later, if that germ comes to town for real, your body can say, hey, I remember you. I'm ready for you now.
Yeah, exactly like that. That's how the immune system sounds. But
like a 1930s cartoon character.
Yeah, absolutely. So most vaccines use a weakened version of a virus or just little pieces of it. But that requires time to grow up a bunch of virus, to process it in order to make the vaccine. But for this vaccine, the mRNA vaccine, there's no virus in it.
Wait. You're saying there's no actual virus in the vaccine at all? Because I didn't really realize that.
Nope.
And so, you have this mRNA vaccine that all you need really was the genetic sequence, the template for what mRNA you're going to make. You didn't need to grow the virus. You didn't need to expose your workers at a lab to it. And so you just skipped a few steps in that way.
That is amazing, Maddie, to hear. It's also a little confusing. Like, what does he mean, the mRNA template?
So mRNA vaccines take advantage of a process that already happens in our body. Like every minute of every day, our body is making this thing called mRNA. And it's like an instruction manual that tells our body how to make proteins. And so if you make a little piece of mRNA in a lab and feed it to our cells will naturally read that little piece of code, that instruction, and use it to make a protein, whatever you want. And in this case, it's a coronavirus protein.
So it's like we're hacking ourselves.
Right.
And instead of putting a bunch of proteins from the virus into the shot, we give the cells in our body the instructions to make that coronavirus protein themselves.
Exactly. That little protein by itself cannot give us COVID. But it is enough to trigger an immune response so that the body learns to fight it.
Wow. So let's say I'm at a doctor's office about to get this vaccine. How do I get it? And how does my immune system actually respond to it?
Okay, so let's walk through it. So these mRNA vaccines are an injection in the arm. After the injection, your muscle cells kind of swallow up the mRNA and bring it into the cell. Now, this mRNA doesn't get into the nucleus of the cell where your DNA hangs out. It can't. So there's no worries about the vaccine like messing with your DNA at all.
Okay.
Instead, that mRNA hangs out in the cytoplasm of your cells where it gets turned into protein.
I remember drawing the cytoplasm in bio class.
Honestly, I think it's an underrated part of the cell.
Yeah.
Okay, so after our body uses mRNA to make protein, the cell puts some of that protein on its surface, like on the outside of the cell, which allows other cells in your body to see it.
And that's the goal, right? The point of the vaccine is for your body to recognize pieces of the coronavirus without actually having to get the coronavirus.
Yeah, and at this point, the vaccine kind of acts like all other vaccines. Throughout the whole process, at different times, our immune system cells recognize and react to that self-made coronavirus protein. I mean, Kwong, there's a whole subset of cells where their entire job is to tattle on invaders. Like, they pick up foreign proteins that are in your body, like this coronavirus protein, and kind of like wave it around to other cells like, guys, I found this protein. It's not supposed to be here. And honestly, I think it should get in trouble.
I'm sure the scientific term for these is not tattle cells.
No, but it should be. Because they're actually called antigen-presenting cells.
Tattle cells is definitely memorable. I'm going to remember that.
Thank you. So these tattle cells can activate other cells, including this diverse group of cells called T cells. So some of them get activated and become very murdery. Those are literally called killer T cells. They go around sniffing out cells infected by the virus and kill them. Some of these T cells help raise the alarm. They bring other cells into the area. I call them super tattle T cells. You know, they're helping with the tattling. And then some of those same T cells help other cells make antibodies. And so later, after you've gotten the vaccine, if you're exposed to the real coronavirus, those antibodies can recognize that protein, grab onto it, and keep the virus from getting into our cells.
And antibodies are what protect us in the long term, right? We want those.
That's a huge part of it. You have this initial wave of antibodies and T cells right after vaccination or infection, but your body also creates this kind of memory bank too.
Once the threat is over, once there's no more presenting of the antigen, the cells will revert into kind of a dormant stage they'll still be around and still be circulating, still growing and dividing until the protein gets presented again, and then they'll activate again almost immediately.
And that memory is why some vaccines work for years and years after you get them. Now, we don't know if this vaccine will lead to long-term immunity, but that's what everybody's hoping will happen.
Sure. All right. Well, I have some general questions left about the vaccine.
Okay, let's do it.
Okay. How long does it take for the vaccine to work?
Okay, so this vaccine is 2 doses, 3 weeks apart. You see some pretty good protection around 14 days after the first dose, but way better protection after the second.
And after the second, can I stop wearing this mask all the time?
No, Kwong. You keep that mask right on. Even in your closet alone right now, I want you to be wearing one. No. At this point, the vaccine has been shown to prevent people from getting symptoms of COVID-19, from, those mildly annoying ones to those really scary symptoms that we worry about that can lead to death. So obviously that alone is huge. But it's possible you could still harbor the virus in your body.
Oh. So I could feel fine, but still have the virus in my body and, I guess, pass on the virus?
Yeah, that's possible. And we will have more information on that at some point. But at this time, it's still really important to wear masks and physically distance with people, especially those who have not yet received the vaccine.
Got it. That's really, really important to hear. Okay. Mm-hmm. And there are side effects to the vaccine, yes? No?
Yeah. Pretty much every vaccine, some people will experience minor side effects like a low-grade fever or fatigue or achiness. And that is normal. It is actually just a result of your immune system gearing up to kind of fight. For some people who have had a history of severe allergic reactions, those people can have bad side effects.
That's why you get asked, have you ever had an allergic reaction to a vaccine? Are you allergic to any of the components in the vaccine? And that's why you look at people and make sure that, you give them the information of what to look for in case they're going to have a bad reaction so they can report back right away and get taken care of.
Scientists are still tracking symptoms of people from early on in clinical trials to make sure that no longer-term side effects show up.
All right. Last question. Mm-hmm. I read that one of the big downsides to these mRNA vaccines is that you have to keep them really, really cold.
Right.
The Pfizer BioNTech one has to be kept at minus 70 degrees Celsius. Why is that? And is that going to be a problem when we're thinking about how to get this to millions of people across the country, not to mention other parts of the world?
Yeah, so mRNA is pretty unstable, which just means it falls apart easily, especially at higher temperatures. So that's why you have to keep it really cold. So, yes, the cold storage requirements are going to put limits on distributing the vaccine, but there is another mRNA vaccine from a company called Moderna that'll be likely approved very soon, and it can be stored at a little bit of a higher temperature, like the temperature of your household freezer. Not to mention, there are other vaccines further down the pike.
That's great to hear, especially since this is a global pandemic. We're going to need other vaccines, some that will use different technologies, some that can be stored more easily to really curb the pandemic so we can take off our masks and live our lives the way we once did.
Yeah, I mean, that is far off, to be honest, and it's important to keep our eye on that. But I do think it's important to celebrate this moment right now. You know, we have to keep an eye on the future and how long this is going to take to kick in, but we also have to take a moment and celebrate the sheer amount of work and investment and scientific cooperation it took to get to this moment, and, you know, the relief that some of our healthcare workers are finally going to have.
Maddie, thank you so much for all of this reporting. It's really fantastic.
Yeah, absolutely, Kwon.
All right. So we just got a dose of the science behind the historic mRNA. What you should have learned is something that we've already covered. To extend that information out, they've been working on the mRNA vaccine for mRNA science for 25 years. And moving forward, they're going to be able to do this a lot faster. Fight pandemics and things of that nature. Also, you learned exactly how it works. You understood that there is no part of the vaccine in there, right? Right? I mean, no part of the actual COVID, the virus in the vaccine, and a lot more. So let's progress forward. And give me a second here. See who joined us. Oh yes, Brother Sutek on the late night tip. What I'm gonna do is I'm going to share this week's Twiv. This is the episode from the other day. Tomorrow with Science with Shawn, we're gonna get into a new episode and something else that I wanted to talk about. Reword the last part. Is there a specific study in the viral dormancy? They studying everything. Period. They got something for everything. Because they doing they working on stuff about the allergies, the allergic reactions some people have had. And they dealing with a whole lot of specifics regarding all kind of stuff. So it is what it is when it comes to that. But I'm gonna show the YouTube video on this one. Y'all know how I'm gonna do. I'm gonna let the talking come in, and then I'm gonna show the video while I'm looking in the chat.
From Michael.
It's late outside. It's a little bright for New York.
Nice.
Yeah.
Very good.
I'm enjoying the sun out here doing Zoom interviews for residences. So
Are you north, middle, or south?
I'm in Sacramento, which they tell me is north, but on the map it looks kind of in the middle.
Oh,
so.
cool. Well, my video guy from ASM, Ray Ortega, is in Sacramento.
Oh.
Very cool. Also joining us from Salt Lake City, Jason Shepard.
Hi, Vincent. How's it going?
Hi, Jason.
How are you? Everything good?
Yeah, pretty good. Actually feels like winter finally, so we've got some snow.
And from Nashville, Tennessee, Erin Calipari.
Hi. We actually got snow too, by the way. Like one flurry one day, but it's been like 75 since then. So that's our winter down here.
75?
It's quite warm. So
You've been away for a few times. Good to see you back. Did you get all your grants done?
I did. I actually got a good score on one of them, so I can't complain. So it's a good day to be back. And now it's the holidays, so things are a little bit more, I'd say maybe less chaotic, but I don't know if that's true. It's a different kind of chaotic. So
Today, of course, first day that the U.S. is immunizing people against COVID-19. It's amazing. It's been less than a year. I'm stunned, frankly.
It's crazy how fast it went. I mean, now we get to look at the logistics and distribution and how effective that is. But the first step of this went really, really fast.
And it worked. I mean, that's the amazing part. It's one thing to plan different vaccines and try them, but they all seem to go pretty smoothly. And as some of you may know, clinical trials are no day at the beach, right?
It's amazing that it's 95%. I mean, I think that was the cherry on the top and how well they seem to work.
Yeah.
And then the second surge also helped.
Yes, we got the results quicker.
Number of cases. Yeah. You
remember for a while we were talking about challenge experiments because we were worried there weren't going to be enough cases, right?
Yeah.
But that wasn't a problem. So, and we have more vaccines to come. There are a couple, 2 or 3 more in the U.S. at least, pretty far advanced. So, could be interesting.
Well, there's the Moderna one, like the meetings with the FDA are this week, right? So
that's right, 17th,
we
right?
To have another approval.
Yeah, and then we have AstraZeneca supposedly, and then Johnson & Johnson and Novavax are not too far behind.
Wow.
And I
Pretty
think
impressive.
It's just amazing, really. From Microbe TV. Johnson & Johnson and Novavax are not too far behind.
Wow.
And I
Pretty
think
impressive.
It's just amazing, really. Back in February, I thought maybe next fall we'd have some vaccines, but here we go. Faith in science, that's what it is. It's all science.
That's right.
A lot of good things ending up.
Faith in science is all science, ladies and gentlemen. All science.
All right, but we're not going to talk about viruses today. We're going to talk about why I can't remember things for long periods of time. Apparently it has something to do.
At least my favorite subject.
And I think Jason's going to lead us through this, right?
That's right. So this is a paper that was published in Nature a month ago called Persistent Transcriptional Programs Are Associated with Remote Memory. And this is from a group at Stanford. This is Tom Sudoff and Stephen Quake. Sudoff won the Nobel Prize a few years ago for his work looking at synaptic transmission. He did a lot of real groundbreaking work looking at how neurotransmitter is released in synaptic vesicles from synapses. So he's a big lab. So this sort of introduces a couple of new-ish terms that I just wanted to start off with, where I think last time we talked about memory, we talked about how there's the structure called the hippocampus that's required for making new memories. And then there's this process called consolidation that can then convert short-term information into long-term storage. And there's this idea that the hippocampus is required initially to start off the cascade of consolidation, but the ultimate site of where information is stored has been somewhat hard to figure out, and in some ways has been controversial. So going back to like the old days where there was 2 camps. One camp is that the information in the brain is distributed across different parts of the brain, that there's no like one site for a memory. You couldn't go in and say, this is where the memory is stored. So that was the one extreme. The other extreme was that, no, there's actual cells that are essential for each memory. And if we just knew where those cells were, we could then reconstitute the memory by just activating those particular cells. So there was back and forth, and I would say that the modern day hypothesis, or the modern day way of thinking is that it's more on the circuit side, that there are specific cells for each memory, but it's still not clear where those cells are.
Right, what components of memory each cell makes up, so it becomes even more complicated, right? Because something that's too good experience is there may be overlapping in the cells that are recruited by positive things, but then there's specific aspects. So it's such a complicated process that we're getting tools to parse apart now, which is why I'm excited about this particular paper.
Yeah, exactly. So this paper is it brings in a lot of the brand new technology that we have for figuring out a few aspects of this memory consolidation process. So that's one sort of new concept here, that the cortex and then parts of the cortex we think end up being where the information is ultimately stored, and that the hippocampus you don't actually need anymore after this consolidation process. So this is called systems consolidation, where the information is ultimately consolidated in a new part of the brain, not the hippocampus, which is where the initial information is processed. And the other aspect of this is that, when we think about consolidation of memory, and we think about, well, I guess the lay public thinks about memory is that, it's some sort of videotape of our experiences. And of course, that's completely false, because we end up changing the memory every time we recall it. So there's subtle updates to the memory literally every time you recall it. And so there's this other concept called reconsolidation, which is that when you recall the memory and there's an updating process that happens, that memory can be wiped out if you interfere with it right at that point. So it becomes this sort of more plastic thing, and then it gets reconsolidated, and now it's now consolidated again into the brain, but with a slight update. And so if you can think about, if you every time you recall a memory, by the 10th time you record the same memory, it's absolutely not the same as the first time.
But this is a really cool process to understand from just a basic neuroscience. How does the brain work? But also, how do we treat disease, right? Because if you think about things like PTSD, where you have these kind of really strong, recurring, kind of inappropriate memories that trigger anxiety responses, how can we use what we know about kind of retrieval and reconsolidation to target those memories and get rid of certain memories that are intrusive while retaining the memories that are really important for our navigation of our environment? And so these studies are so important for just how does the brain work, but also how can we kind of harness this to really treat this in disease, but also not have adverse effects. So that's why this is so exciting, right? There's just so many levels for, if we can figure this out, how we could use it in disease and all kinds of things like that.
Yeah, exactly. So one idea is that PTSD is this, super salient memory that's tied to an emotional content. And perhaps one way to get rid of the emotional side of that memory is to have people recall the memory in a safe context, a safe environment, so that over time that memory gets updated, but you sort of start to lose the real strong emotional content. And that has panned out in human studies, where some of the therapies that people use for PTSD are exactly that you sort of get people to recall the really painful memory, but in a very safe context, in a safe environment.
Jason, in the humans, how long does it take for this memory consolidation? And does it require, so the original thing that you're remembering, do you have to repeat it, or is it enough to do it once?
Yeah, those are all good questions. So there are some kinds of memories that you only need one event. So, and that's usually these emotional kinds of memories where you can imagine that they stick in your mind, partly because they elicit such a big emotional response. For other things that are boring as hell, like trying to memorize, math equations, you do need repetition. So the brain is wired to consolidate things that are going to elicit an emotional response, because of course in the environment there are these stressful things that you would want to learn quickly are things like, where's that predator, or I ate that bad food and I don't want to eat that again.
It's all about survival, right?
Right. Exactly. And in terms of the timing, it's not actually that clear, but I would say, generally speaking, this molecular consolidation happens within days after learning, but the systems consolidation, where the ultimate storage in the cortex happens, takes about 3 to 4 weeks, so about a month. And so in this study, and again, this is a study in animals, so they're using mice. Their setup is their behavioral setup is this fear conditioning memory. So they basically train the mouse with putting them in a specific context in a box, and they shock the mouse, a mild shock, enough that the mouse obviously doesn't like it. And then a couple of trials, it doesn't take that long, because this is the kind of memory that does elicit emotion in a big response. The training is pretty short, so they only take a day for training, and then you can get a robust memory that lasts for months. And the consolidation, so then they basically take the animal after learning, leave them alone for 2 weeks, 16 days, and then put them back in that same box that they were shocked in, where they know that the animal is going to recall that memory. And at that day, 16, they use some fancy genetics in the mouse to mark the neurons that are active during that recall event, with the idea that those active neurons that are being activated during the recall event are the neurons that are part of the consolidation process, or the reconsolidation process. And then they come back 9 days after that recall event and actually take out those cells. So they've used the mouse genetics to express a fluorescent protein in those active neurons. And then they sort those neurons 9 days later out of the mouse brain and use the latest RNA sequencing technology, single cell sequencing, to see what gene expression changes have happened during the 9 days after the recall event in this behavior. So it combines, and I don't think we need to go into the details of how they trap the active neurons, but basically it's using these genes that they know are active, are activity dependent, that drive a reporter, and that is controlled by other ways of sort of keeping the reporter only active.
We work with this mouse line, so it's incredibly hard to explain. Yeah, cells that are on, those are tagged with a fluorophore. So the cells that were active are red in their case. Yeah, it's a funny mouse line. I think the one thing that's really interesting, that's really cool about it, is you're tagging the cells that were activated during some window, right? So it's not just defining cells by some genetic thing they express, it's what cells are active. And it gives you this kind of cool window into not just it's glutamate or GABA cells, it's the kind of ensemble, or as they call them, engrams, of neurons that are all co-activated. The one downfall of this approach is that you're labeling cells that are activated. So if you have some neural process, which we also study stuff like this, that is encoded by a reduction in activity in a specific population, you're not studying those. And so this is kind of cool because you're saying, okay, which cells are repeatedly activated and recruited, and you're kind of ignoring the stuff that's shut off. But in my opinion, kind of both of those processes, I don't think that you need to study both of them in every single study, but both of those are really, really important. I think sometimes the neuroscience field forgets that getting rid of neurons, like in development, when you have pruning, is sometimes just as important as activating things later, and kind of that yin and yang balance is such a really critical part of information encoding. But this paper is cool because it looks at what the transcriptional programs are that cause the persistent recruitment of the activated population, which is really critical for these memories.
You could look at the ones that go down, right, in a different experimental approach, right?
Yeah, it's kind of hard though, because then you'd have to have things. So we do this, and we're funded for this. You need some like double labeling approaches. So you have cells that were like activated before and now are no longer activated, which is hard. But with single cell sequencing, we're able to do that because you have they're tagging their cells with a fluorophore, but you also.
I know I'm boring some of y'all, but the reason for this is because the first part of this is important, because they're trying to use some of their understandings to corroborate that information and intel into the second part of the discussion, which is like future research and things of that nature. However, the key word that is being used here is cells. Cells. When you're dealing with the mRNA vaccine, you're dealing with something that is dealing with cells.
Also get the transcriptional signature of those same cells. So if you tag something 6 days ago and it's no longer activated, you should be able to tell that in the transcriptional readout of the cells based on activity markers and things like that. So with the big data in this, it's just like we have like unlimited possibilities with what we can look at and how.
The other major caveat here is that they're using one gene called FOS, or the promoter for that gene, to mark these active neurons. And there's, I would say, there's some debate on, are you capturing every single event or every single kind of active cell with this one promoter? But let's say you can capture most of them with this promoter.
Yeah, and what patterns? My husband's a scientist, and he does like 2-photon microscopy. So we've been having this, I'm gonna say, dinner argument about in these mouse lines what activity patterns do cause transcriptional activation. So is it a single action potential? Is it repeated phasic firing over long periods of time, and kind of what is the recruitment you're getting? And so we've been talking about doing some of those kind of low-hanging fruit experiments because we can record and tag cells at the same time, and with some of his fancy microscopy tools to see if, what patterns are being recruited. Obviously, people have shown these are involved in the memories, right? Because you lesion them, the animals don't have behavioral responses. So there's a lot of evidence these are critical. The question is just kind of in what way and what is recruitment, which is
time to do those experiments. You guys should do those experiments. I think the more of those kind of experiments that are done, the better now, because there's, like I study this one gene called ARC, which is also activity dependent, but it labels a slightly different population of cells when you look at them and you look at the overlap with FOS. So there's subtleties here. But
that's the plan. So we have the ARC driver lines and the FOS driver lines, and to see if different activity patterns recruit different cells in these different mouse lines, and if there's overlap and what you're really looking at, which I think is just one of those basic questions about the genetics that it's just been hard. If you don't have the tools to do it, it's not easy. So this is my fun Christmas project.
Yeah. Sounds easy. All right. So we've got these mice. We've got these various groups that they use. So there's the untrained groups, so they don't get paired with the shock, but they see the same context. And then they have, and that's really the main control, that everything, the experience that the animals are having are almost exactly the same, except they never got trained with the shock. So they're seeing the same context, and they're getting, and you can imagine that, 16 days after the first event or the first time they are exposed to this, if they're not shocked, they may actually see this as being a new context.
And so there's a lot of kind of, it's really hard when you're doing like this isn't about that, but we do sex differences stuff sometimes, how these tools can also on their own affect transcriptional regulation in the animal. Here they have controls, right? Because their negative cells are coming from animals that also got this drug. So it's not like you can attribute all of this to that regulation. But
all right. I ain't gonna bore y'all with the rest of this, so to help y'all understand mRNA. But basically, the root of this is that the mRNA for long-term memory, right? They're dealing with this from a perspective to dictate or see what the long-term memory would be, but they're using animals right now. They're testing this they're testing animals to understand how the mRNA for long-term purposes is going to behave and act. So you're hearing a scientific conversation about the study of cells related to these particular animals and their behaviors and things like that affect them long term, if there's any type of affection long term or whatever that needs to be recorded, so that that data can be transferred into a human trial, which will later help us better understand how mRNA vaccines are going to do so much more for us. I'm gonna kill it right there. I'm gonna stop sharing my screen for a second. I know I lost a lot of y'all with this particular conversation, but we're gonna get into a bite-sized piece of what we're going to do tomorrow. But I wanted y'all to know pretty much what they're dealing with when they're regarding the mRNA. Straight up, because that's what we're dealing with today. And that's what's going down. So again, I shared a link last time, man, about that assistant manager's position that they were actually trying to throw out there. And we'll kind of go from there. Give me 1 second, and I'm gonna get into the brief preview of tomorrow's show. We'll get into a little bit of stuff. The Raiders finna lose. Almost. And we're going to get to, let me get to 17-minute mark. And then I'm going to share. All right, so this is what we're going to be talking about tomorrow. And shout out to everybody, man. I just seen the attendance go up, man. We appreciate everybody hopping up in this joker, bro. But we just dealing with some science. It's science with Shawn. We getting it in. I'm gonna take it back just a little bit so we can segue right into the discussion. Let's see what these scientists talking about.
Not a bad guy. He's just caught between a rock and a hard place.
Yeah.
Right? Because, if he doesn't do what the administration asks, he gets fired. And maybe he wants to be able to affect change, as Kathy said about these employees, right? If you think you can make it better. So there you go. All right. This week I was looking at the influenza statistics. And it was quite interesting. You can go to CDC FluView, where you get weekly statistics. These are influenza positive tests reported to the CDC by U.S. clinical laboratories all throughout the country. Wonderful statistics. And this is week 49. And you can see we've had a number of weeks of reporting so far. And the numbers are quite low. The graph is pretty flat. There are 40, in week 49, there were 40 positive specimens out of 22,474, 0.2%. And last year at week 49, I went back, and you can look in the archive for the information. Week 49, there were 3,436 positive specimens out of 30,000, which is 11.3%. So it's a big difference. And that's clearly, well, one would hope that it's a consequence of the non-pharmaceutical interventions, masking and physical distances, NPIs.
Yes.
However, if you go to the CDC site, they say these data should be interpreted with caution. I don't know who's actually written that, right? In light of our conversation, because sampling is off this year, right? And, there are 22,000 specimens compared to 30,000 in week 49 last year. It's not so far off, but maybe it's skewed to places where, so I don't know.
Right.
But
it's 86-fold lower.
Yeah.
That's a lot.
Yeah, it's really impressive. My pick that we'll get to later sort of gets at some of these same issues. And I have just been so impressed by this and think that we need to be talking about this more. I remember a lot of questions people were asking me at the beginning of the pandemic. And I think that this addresses some of them.
Well, I think you cannot prove that this is from the COVID efforts, right? You cannot prove it, but it certainly strongly suggests at it, right? And I'd really like to know if CDC thinks that instead of saying, interpret with caution. I don't know why that is.
I'm sure this will be a subject for future investigations. We're going to hear a lot more about this.
Oh, certainly.
But I'm going to keep track of this and see every week what's going on if it stays flat, because usually now it starts to go substantially for the next month or two. And if it stays flat, that's amazing, right?
I wonder what the uptake on the flu vaccine has been this year.
Yes, you can find those numbers at the site. I didn't look into them, but they do measure the distribution, not whether it's actually injected into arms, but the distribution.
It'll be interesting to see if you see a bump after Thanksgiving, for example, or after Christmas holidays, if there's enough granular information there to see that.
You can go and you can see by state, the positivity rates. So very nice data. All right, and the other issue, of course, tomorrow, Moderna's vaccine results are going to go before the FDA. They'll probably issue an EUA on Friday, as was done last week for Pfizer. I love it, Tony. Pfizer. Tony Fauci says Pfizer. And Charles sent us a link to the documents that are part of the meeting that all the members of the panels are going to look at. And we'll put a link to that. There are 7 different PDFs that you can look at. Lots of data there. And this is good because it'll bring more millions of doses to the US, which is great.
Yeah, so there's some really fascinating data that is a part of this, both in terms of the efficacy of the vaccine, but also with one of the questions that we've been asking quite a bit has been about prevention of infection versus prevention of transmission. After the first of the 2 doses, this is a vaccine that has 2 doses, they did do nasal swabs. So they can start to assess whether infection is being prevented. Of course, they only show these data after shot 1. They don't show it to us after shot 2. But the data indicate, I believe it was a 67% protection from infection, while it was a 94 or something percent protection from disease. They do see some protection from infection. There's a different number of infections in the PCR positives in the placebo and in the vaccine group after the first administration.
Yeah. And the Pfizer we talked about last week was like 50-something% protection in the vaccine group against asymptomatic infection. And that's better than nothing, but it leaves a lot of people still susceptible to infection. Not serious disease. The 94 is the serious disease prevention. That's great. But this is a concern because a lot of people think that they are going to get the vaccine and rip off their mask and go back to life as usual. But you're going to have to wait for more people to be immunized for that, because 40% of the people immunized are still going to be able to transmit, be infected at least, and maybe transmit. We don't know.
Brianne, do you know if they're doing the swabs after the second shot?
I didn't think they were. I thought they were doing blood samples, but I
I'm gonna do this again because I really think that y'all need to understand what is being said right here.
Part of the meeting that all the people, members of the panels, are going to look at, and we'll put a link to that. There are 7 different PDFs that you can look at. Lots of data there. And this is good because it'll bring more millions of doses to the US, which is great.
Yeah, so there's some really fascinating data that is a part of this, both in terms of the efficacy of the vaccine, but also with one of the questions that we've been asking quite a bit has been about prevention of infection versus prevention of transmission. After the first of the 2 doses, this is a vaccine that has 2 doses, they did do nasal swabs. So they can start to assess whether infection is being prevented. Of course, they only show these data after shot 1. They don't show it to us after shot 2. But the data indicate, I believe it was a 67% protection from infection.
67% protection against infection. And then you have a 94% efficacy rate against. So what they're talking about is between shot dosages. So they cotton swabbed between shot dosages, right? And this is what they're trying to do. They're trying to work on a prevention measure all the way around the board. So this data is valuable, because if you got millions of people that has vaccine 1, and you remember they're talking about they were looking for an efficacy rate against the virus of about 60%. You get that at 94, but around the board in between shots you're at 67. But we really don't know because there's more data to come out. You know, this is going to answer all the questions. So not only are we asking the questions, right? Scientists are asking questions, and we're getting more data as we go along with this, which helps us all in the long run. But just let's hear out.
While it was a 94 or something percent protection from disease, they do see some protection from infection. There's a different number of infections in the PCR positives in the placebo and in the vaccine group after the first administration.
Yeah. And the Pfizer we talked about last week was like 50-something% protection in the vaccine group against asymptomatic infection. And that's better than nothing, but it leaves a lot of people still susceptible to infection. Not serious disease. The 94 is the serious disease prevention. That's great. But this is a concern because a lot of people think that they are going to get the vaccine and rip off their mask and go back to life as usual. But you're going to have to wait for more people to be immunized for that, because 40% of the people immunized are still going to be able to transmit, be infected at least, and maybe transmit. We don't know.
Brianne, do you know if they're doing the swabs after the second shot?
I didn't think they were. I thought they were doing blood samples, but I cannot say that I am an expert on all 90-some pages here.
Because this is, whether or not they're doing it as part of the trials, this is a critical question.
It really is.
I would imagine that somebody is following this up.
It is critical, but the primary and secondary endpoints were both disease.
Right.
Right. Because it's hard to do this, and it's probably self swabbing as well, right, Brianne?
Yeah, I believe it was self-swabbing. Of course, I think as we mentioned before, they have been taking blood samples, and so they could look for immune responses to antigens not in the vaccine and sort of get an idea about infections. But I do really like the data coming from the swabs.
It's hard to prevent infection. 60-something. What would you just say? What was the number? 60?
I believe it was 67.
67 versus 94. Those numbers are still small, right?
Yes, the numbers are very small.
Okay.
And again, that was only after 94 is protection from disease after 2 administrations.
Yeah.
The 67 is protection from infection after 1.
Very important question. As I said in the pre-show is before we start recording. You never hear it. We talk a lot, sometimes too long. I had a doctor's appointment this morning. I was sitting in the room and I have the goddamn TV playing, which I don't want because I hate TV, but the news is on. They're showing the first people get immunized. Okay, great. That's great. Then the guy says, this vaccine is 94% effective at preventing infection. No, it is not. It's 94% effective at preventing disease. As Brianne just told you, it may be 68% effective at preventing infection after the first dose, but we need more data on that. But, they don't, I don't know why. Why do they do that?
Why? Because they don't understand the difference.
Right. It hasn't been pointed out there's a difference in how you say it. Just like a lot of people don't understand there's a difference between social distancing and physical distancing.
There's a certain kind of, I don't know that it's carelessness, but not really understanding all of the dynamics. This is what Twiv is for. Okay?
Yes, I was going to say, there's no excuse not to know this. All you have to do is listen to Twiv. Okay. Now I know it's long. Maybe you want me to go through and extract every time we said this and put them all in one long episode where we say over and over, vaccines don't prevent infection, over and over.
Infection does not equal disease. Infection does not equal disease.
Listen to Twiv, folks. Can I rant for 1 minute?
Oh, good. Take two. Go for it.
All right. I'm timing you.
I looked this morning on YouTube. There's one video called Ivermectin and COVID-19, and another video called Vitamin D and COVID-19. They both have over 150,000 views because people want to know about some easy way out of this, right? And we never get those many views because we talk about science in a long form. So I think that's part of the problem. You don't want to learn. And all you listeners, you want to learn. That's why you're here. But
see, here's the challenge, right? And I think this is critical. I wish Uncle was up. He might be asleep. But we're having a bigger issue in our community because nobody wants to learn. But they're also facing the same challenges because Twiv isn't worldwide or on a scale that it needs to be among people that they converse with. But here we are. He's talking about, does the virus protect against X, Y, and Z? And then Brianne turns around and she gives it the most simplest form and says, infection does not equal disease. So you can get the vaccine and still be infected, but it will not equal the disease. And we missed that. But I mean, just look, you had people, and I guarantee you that the video that he's talking about regarding vitamin D and over the vaccine, and they got a bunch of views, is because Black people probably tuned in to that. One in particular, Ali Muhammad, because that's the same mess that he was kicking right in there, talking about vitamin D. He was kicking that. Vitamin D ain't gonna prevent jack. So let me rewind it back, man. Watch his facial expression. Watch Vincent's facial. Look at his face right now. But let me write just a little bit more back. Let me get to it.
I didn't think they were. I thought they were doing blood samples, but I cannot say that I am an expert on all 90-some pages here.
Because this is, whether or not they're doing it as part of the trials, this is a critical question.
It really is.
I would imagine that somebody is following this up.
It is critical, but the primary and secondary endpoints were both disease.
Right.
Right. Because it's hard to do this. And it's probably self-swabbing as well, right, Brianne?
Yeah, I believe it was self-swabbing. Of course, I think as we mentioned before, they have been taking blood samples, and so they could look for immune responses to antigens not in the vaccine and sort of get an idea about infections. But I do really like the data coming from the swabs.
It's hard to prevent infection. 60 something. What did you say? What was the number? 60?
I believe it was 67.
67 versus 94. But its numbers are still small, right?
Yes, the numbers are very small.
Okay.
And again, that was only after 94 is protection from disease after 2 administrations.
Yeah.
The 67 is protection from infection after 1.
Very important question. I mean, as I said in the pre-show is before we start recording. You never hear it. We talk a lot, sometimes too long. I had a doctor's appointment this morning. I was sitting in the room and I have a goddamn TV playing, which I don't want because I hate TV, but the news is on. They're showing the first people get immunized. Okay, great. That's great. Then the guy says, this vaccine is 94% effective at preventing infection. No, it is not. It's 94% effective at preventing disease. As Brienne just told you, it may be 68% effective at preventing infection after the first dose, but we need more data on that. But, they don't, I don't know why. Why do they do that?
Why? Because they don't understand the difference.
Right. It hasn't been pointed out there's a difference in how you say it. Just like a lot of people don't understand there's a difference between social distancing and physical distancing.
There's a certain kind of, I don't know that it's carelessness, but not really understanding all of the dynamics. This is what Twiv is for. Okay?
Yes, I was going to say, there's no excuse not to know this. All you have to do is listen to Twiv. Okay. Now I know it's long. Maybe you want me to go through and extract every time we said this and put them all in one long episode where we say over and over, vaccines don't prevent infection, over and over.
Infection does not equal disease. Infection does not equal disease.
Listen to Twiv, folks. Can I rant for 1 minute?
Oh, good. Take 2. Go for it.
All right. I'm timing you.
I looked this morning on YouTube. There's one video called Ivermectin and COVID-19, and another video called Vitamin D and COVID-19. They both have over 150,000 views because people want to know about some easy way out of this, right? And we never get those many views because we talk about science in a long form. So I think that's part of the problem. You don't want to learn. And all you listeners, you want to learn. That's why you're here. But everybody else, where are they? Why don't we have 150,000 listens or views? Because I can guarantee you that you can't use ivermectin for COVID-19. Vitamin D may or may not work. Just take it, as Daniel said. Just take it. Why have a whole podcast about it? Okay.
It's hard. Learning is hard,
There is no expedient.
You have to be curious, okay, to make it easier. And a lot of people just want the answer,
You have to admit you don't know something in order to learn.
Yeah.
Right.
Yeah. We have a very interesting paper, which will, the reason I picked it's hard paper. I had to read it multiple times, and there are no pictures. Because 2 main points. One is if you don't do mitigations like non-pharmaceutical interventions, NPIs, this virus can rip through a population and cause a lot of trouble. And so, people have like the Great Barrington thingy, right? They say, just let it rip. And you can't, because then it affects a lot of people, as you will see, because that was the situation in Brazil. And secondly, there are a number of, they use serological surveys, and we're going to see how you can use those data, their limitations, and figuring out the infection fatality rate, which is different from the case fatality rate, as we'll see. So those are why I think this is a cool paper. 3/4, it's published in Science, 3/4 attack rate of SARS-CoV-2 in the Brazilian Amazon during a largely unmitigated epidemic. They didn't do anything.
The article he's talking about right now, I actually had posted it on Facebook, but I will make sure to add it in the description for this one. Because it would, for those who want to get involved with it. But I'm going to stop right here because I don't want to expound my video too much longer on that. And before we like end the bill, I'm going to try to find this video too on Twiv regarding the regarding the milk and stuff. So well, not milk, vitamin D, sorry. Vitamin D. Right? just so people can have it, because this was a big deal about a month ago, and this is right around the time y'all think Ali was on or something. A month ago, they was talking about the vitamin D and the vaccine. So when he came on with his whole video toward the end, a couple of weeks ago, this was a story that was out a month ago, and he know people probably wasn't paying no attention to that. No attention to it. So I'm gonna try to look for it, but I hope everybody understand what the sister was like and what he was saying about infection, preventing infection, and an infection, dealing with the disease. I hope y'all kind of get out of that, because there's a lot of work being done by these scientists, and they're researching, and this panel that we were just listening to, they're not doing anything different. And these scientists that are on that panel, oh yeah, they scientists, they qualify, they in the field, they know the studies, they know what to look for. They're part of probably these peer review panels and more. But what they're doing when they're getting the data from Moderna or the data from Pfizer, they're breaking it down like me, you, and others are. It ain't no special recipe. The difference between them breaking it down and us is they understand word for word everything that is being written. Those big words we can't make no sense out of, they make all the sense out of it among themselves. They crack their little corny jokes and they keep it moving, but they are trying to scientifically move the people in the right direction based on the data. And when they have questions, they pose a question, all of them, and then they say, well, we will need this data regarding to this, or yeah, I can't wait to see what the data is showing on that. They know people are in these rooms asking the same questions that they're asking one another on a panel and dealing with the science. But no way possible. You heard him say it. He was like, man, we should be getting 150,000 views on our stuff. And they getting 20, 30, 40, sometimes 80. You know what I'm saying? Their most viewed video, I think, got everything to do with it, with the vitamin D. But yeah, I'm trying to look in the chat real quick, see what y'all on, man.
Yeah, man, I think it's key. I was just saying, I think it's key to know these terms and these scientific concepts. That's why I'm glad you played that over a couple of times so people can catch that, because I know a lot of people, they hear something and they hearing it in layman's terms. You know what I'm saying? It's their scientific terms and concepts, but it's going in your ear, not the other, as layman's terms. So it's good that we learn these scientific terms, man, because we can understand a lot more of what's being said and what's being talked about, and not being left in the dark and ignorant.
Facts. So I'm gonna go back to this video, right? And I just want to show people what I'm talking about. Now, Ali video was on like the 27th or 28th. It was around Thanksgiving, right? Or right after Thanksgiving Day. This was out on October the 4th. Let's see what they're talking about right here.
Viruses, the kind of activities, and I'm going to make sure I hit this here, but yeah, that's Pfizer and Moderna may, right about the end of October, beginning of November, start to have some of the preliminary efficacy data. It's not safety data. That's not saying let's rush out and get vaccinated. But and then we think with the Johnson & Johnson, it'll be late November that we start to get some of the early efficacy data.
And you said we need 200 infections to start making efficacy data, right?
That's the goal in these studies is to 200 infections. But early preliminary stuff, you're going to start seeing trickles early on with less than 200 infections. And we talk, and I'm going to make sure I hit this here, but yeah, that's what they're talking about. They're talking about less than 200 infections, but starting to say, IUs per day. And when you get above 70, we recommend about 800. But if you're deficient, it may take a more significant amount to get you up into the normal range.
It's a good,
so for those of you that want more about vitamin D, we need more research.
There's a good summary in Lancet Diabetes and Endocrinology, and they point out it's been thought for years that it might have a role in respiratory infections.
Cases still open. And so I want to actually say, as excited as people might get about vitamin D, you want to be cautious here, where the jury is still out. And again, this is a fat soluble vitamin, so you want to be careful here. Generally, someone should not exceed 4,000 international units per day, unless specifically recommended by a physician. Right? That's the little disclaimer they always have on those commercials. Because if you take too much, as I mentioned, you can get sick. You can get hypercalcemia. You can start peeing out a lot of calcium. You can actually start peeing out a lot, which then you end up needing to drink a lot. You can get confused. You can vomit. You can actually have bone demineralization. It's not like, more is better. And so, if you're interested in getting your vitamin D checked, we actually have a huge vitamin D deficiency problem in the United States because, like Vincent, if people are watching our thing, he is fully clothed despite the fact that this is a beautiful sunny day. He's indoors, it makes it worse. So yeah, we're not spending that time that we spent outdoors as humans evolved. We're not getting our sun exposed to UV, our skin exposed to UVB. We're not getting hot. We're not making that amount of vitamin D. So when they do surveys in the United States, particularly as we go into the winter months, vitamin D deficiency is a broad issue. And if this is related to COVID-19, that's not positive. And so again, it's COVID-19. It makes sense to exercise, makes sense to eat right, makes sense to have an ideal body weight. And if your vitamin D is deficient, I don't think there's a problem with getting it into the normal range. We don't know if that will help, but don't get it above the normal range. So, for people under the age of 1 year, I don't know how many listeners we have, but that's about 400 IUs per day. Generally 1 to 70, it's about 600 IUs per day. And when you get above 70, we recommend about 800. But if you're deficient, it may take a more significant amount to get you up into the normal range. So for those of you that want more about vitamin D, we need more research.
There's a good summary in Lancet Diabetes and Endocrinology, and they point out it's been thought for years that it might have a role in respiratory infections in general, and in only one randomized clinical trial did it show an effect. And they basically said everything you have, and that there's a lady in Trinity College who says, the circumstantial evidence is strong. We don't have a randomized controlled trial, but how long do you want to wait in the context of the crisis? We know vitamin D can be good for you. So as long as you don't overdose, why not take it? It seems to be a reasonable, that's what you're saying basically, Daniel, right?
Yeah. Yeah. And with that caution, I think don't overdose. Go see your doctor, get your physical, get your vitamin D checked. If it's low, get it into the normal range. If it's high, rate it back. All right. Vaccine.
All right. So real quick. Now, if any of y'all that watched the video with Ali Baba Muhammad Amuru Yakum Shawa, y'all all heard him talking about when he got to talking about vitamin D that they were trying to keep us in the house to get us ready and prepared for the vaccine. But here we are with the vaccine scientists and the doctor right here, right? We dealing with Dr. Griffin, right here. So we're dealing with Dr. Griffin, and Dr. Griffin is telling us off the top that all people have a vitamin D problem. And Ali tried to make it seem like we so different than everybody else that we don't have a vitamin D problem, but it's going to cause us to have a vitamin D problem because we inside. Yeah, Pure said Bernstein is the boy, you crazy. I agree. I agree with that, Pure. You're right. But nah, let them build. So let them build. You know, I was gonna end it, but you know what I'm saying. We got a couple people in here. Look at that. Trump main driver code misinformation. We're gonna skip to this in a minute. But let me see what he's saying. Nah, you know what? He finna get rid of the switch lanes. Finna talk about Johnson & Johnson, and I don't really want to get on that. But I will put this episode in the chat for anybody who's interested in want to come back to that information.
And they'd been in groups at different times. And so I think the contact tracing might be a little bit of a challenge, given some of the groups that had been together. But I am not an expert in contact tracing to be sure about that.
Well, I just updated my New York Times front page here, and it has that Mr. Trump and Melania and Hope Hicks have tested positive, that Pence, his wife Ivanka, Jared, Barron Trump, Alex Azar, and William Barr have tested negative, as has Biden. Okay, so we're going to get reports on some of this, but with respect to the sort of analysis that Joseph is looking for, regardless of what might be done, I don't trust anything that comes out of that office. Nothing.
Interesting. By the way,
I do not trust it.
One of his aides,
rightly so.
Hope something. What's her name? Apparently Hicks. She apparently was positive first, and he knew it. And was hanging out with her, and probably got infected by her. And then he went to other meetings without even worrying about it, without wearing a face mask or telling anyone.
Vincent, he doesn't believe that this is an infectious disease, apparently, because you can watch him at rallies. He doesn't think anything bad is happening at those rallies, and he's absolutely dead wrong. Yeah. No pun intended.
Well, speaking of the New York Times, there's an article here. Study finds single largest driver of coronavirus misinformation, Trump. Cornell University researchers analyzing 38 million English language articles about the pandemic found that Trump was the largest driver of the infodemic.
Correct.
And there you go. Now he's infected.
Correct.
Do you think it will change? Do you think it will change any of his attitude about the pandemic?
If he gets seriously ill, it will. Mm-hmm. If he's one of those non-responders and he gets, he's an asymptomatic carrier, then the answer is no.
And if he does not get seriously ill, okay, he'll say, "This is nothing."
He was tough. He was a tough guy, and he fought it off.
Dixon, can you?
Yeah, it would be nice if it did change, simply because that would save a lot of lives.
Can you move the mic a little bit away from your beard, please?
Oh, I'm sorry
rubbing it a little. That's
how
good.
Is that?
Thank you, sir. Thank you, sir.
You're quite welcome, sir.
What did we just say?
Well, it depends on how sick he gets as to whether he comes clean or not.
I think Daniel's right. If he gets remdesivir now, this is going to knock the virus, right in its tracks. Remdesivir early will do it. Okay. There is a very good article in The Scientist, which I recommend to everyone. You can easily read this. It's about the Surgisphere scandal, and it's really, really very well written. Okay, this is my browser is doing weird things. Excuse me while I refresh it. Okay, the Surgisphere scandal. They had done a big study of the use of hydroxychloroquine, right? For treatment of severe COVID-19. They pulled together 100,000 medical records across 671 hospitals in 6 continents. And this was some time ago in May. And based on that report, Trump said we should use it, and people started using it. And it turns out that that whole study was bogus, most likely. And they've investigated it. This company is pretty weird. It was started by a surgeon, apparently, and had various incarnations. And then it did this big study. And so basically it's been accused of fraud. And this article details it. I don't want to go into the details, but I highly recommend it.
Central to this, and this was news to me, central to this is what he calls a proprietary patient database.
Yeah.
Proprietary patient database from which he drew all the data for his conclusions. And the people who initially raised a flag about this said, wait a minute, there isn't enough data in the known universe to populate this database in the way he says it's populated.
Right.
That's right.
And the thinking is now that, in fact, the whole database is fraudulent.
It's bogus, yeah.
Bogus.
This is just horrible because this is people's lives we're dealing with. Why
what
would
does
you do this?
Surgisphere refer to, Vincent?
It's a company.
It's just this guy's name.
Oh, that's the name of the company. Oh,
Yeah.
okay.
Yeah, it was founded by
Okay.
what's I'm sorry, Sapon
Surgeons. I see,
vascular surgeon
surgeons.
Sapon
I got it.
Desai in 2008. Initially produced textbooks and later dabbled in various projects, including a journal, before shooting to fame this year with his high-profile publications on COVID-19. So,
And he has a very checkered career.
Vincent, you have an absolute right to ask, why do people listen to that SHI, what, rather than us? There's no accountability in something like this. We take our marching orders from our listeners.
Well, look,
if we screw up, we learn
we
screwed up.
We get called out, but also we call out each other if we say something wrong.
Of course, and it's transparency that matters most in all of this, right?
Yes, of course.
So remember, don't trust scientists, trust science.
But the other thing is, Dixon, we're not doing this for money, right?
No, that's true.
We're not being paid. There's no wealth aggrandizement. We're a Surgisphere.
Would it matter if we were?
No, it wouldn't. But people could then say we're doing it for the money. People would say we're doing it for the money, but that card is gone. They can't use that.
Really,
right?
you can't live on the money that we've generated. Trust me.
So
it's sad. It's very, very sad.
Don't trust the scientists. Trust the science.
We do it.
Well, it's important to remember, as Dixon says, don't trust science, not the scientists, that anytime there's anything like this going on in any area of science, there's going to be a few bad apples.
Absolutely.
Who, wind up getting a lot of press because they make a lot of noise and say things that people want to hear. And you have to learn how to, parse the good away from the bad.
Yeah, the biggest thrill a scientist should get from making a discovery is the discovery, not from the recognition of the discovery.
For sure.
And that's the difference between scientists that get carried away with their hypotheses and scientists like Vincent and Brianne and Rich and myself, that we glow when we've discovered something new and you keep moving on.
You remind me of that favorite, one of my favorite sayings, I forget where this comes from, is that the most impactful phrase in science is not eureka, it's, oh, that's funny.
Exactly. You got it. What? I thought it was.
All right. I want to discuss briefly a preprint on bioRxiv that just was posted on the 29th of September, which bears on a arc we've had here on Twiv. And the paper is called SARS-CoV-2 D614G variant exhibits enhanced replication ex vivo and earlier transmission in vivo. And this is a collaboration between laboratory Barrick and Kawaoka laboratories, University of North Carolina and University of Wisconsin-Madison. And as you recall, D614 is a single amino acid change in the spike glycoprotein of SARS-CoV-2 that arose early and has since predominated, and it has been suggested. All right, so there's one experiment where they have 5 of 8 animals on day 2 are infected through the air, presumably with the variant, but not wild type virus. And they say this arose early on and is just transmitted to everyone.
Yeah, sure.
Which stands for.
Putting me on the spot.
No, it doesn't
again.
stand for, but this is Dixon, not Dixon. Daniel, he says he remembers it because it's old cow. Okay, sorry.
Right,
All right.
remember it.
So, we don't know, but I think that this is a really interesting idea.
Yeah.
Because
Norway.
Think so?
I don't know.
I'm trying to get to the Q&A. I want to see if they're going to talk about something else.
Because that's the one I thought they engineered to, it could be quite helpful in public places. I'm not sure how I feel about common use in private homes over extended periods, particularly when those homes hold
completely sterilizing and giving an immune system little exposure. I'm pretty sure your immune system gets quite a bit of exposure. I suppose if the sterilization was amazing and got rid of absolutely everything, then I might worry. Make some joke about how their mom didn't let them eat enough dirt.
On different skin areas, and it does, of course. Could you?
All right, so they ain't do it on this one. They usually got it going on inside of there. Where we at 2:40? Okay, with 20 minutes left. Let me see.
That's the point. Right. All right, Igor writes, Dear Vincent and friends, regarding your discussion of the Russian SARS-CoV-2 vaccine, I want to make 3 comments. First, you did not emphasize this, but the trials described in the Lancet article can hardly be described as phase 1/2. I agree, but that's what they did. They described it as the liquid vaccine and the lyophilized vaccine have to be treated as different products because they follow different manufacturing processes. Thus, one should not bundle the results of the 2 together. And the actual trial of each vaccine involved not 76, but only 38 people. Of these 38, 9 received AD5, 9 received AD26, and 20 received both. Authors call the first 9 and 9 as phase 1, but that is not accurate. Since their final vaccine product has both AD26 and AD5, these first 18 people should not be called phase 1 trial of the vaccine. The 20 people that got both AD26 and AD5 are the true phase 1 participants. To be clear, the vaccine was, quote, licensed, unquote, based on the data from these 20 volunteers, which barely qualifies as phase 1 and is certainly a major stretch to be called phase 2. Second,
That's too much. All right, so I left a video for that in the chat. I also left anybody that want to read this particular article or have any type of interest in what he was talking about regarding the D614G, surrounding the S protein. If you're familiar with all of this, you're starting to see things that we've dealt with before. ACE2, transgenetic, D614G. Really, the keywords like this, S protein. Of course, we should know what this is. Right? These are things that we're seeing previously on other episodes. And we continue to learn and continue to learn and continue to learn and continue to learn until we know their lingo. And then when we know their lingo, then going through this right here becomes even more easier for us, right? Because they give us a glossary so we know exactly what they're talking about when they're abbreviating these things. You heard me keep saying the EAU, that's emergency use authorization. We've been hearing it. We're going to continue to hear EAU regarding all vaccines that come aboard. Even and what people don't know is that, yeah, the mRNA vaccines are coming first, but behind it is some conventional vaccines. Potential conventional vaccines, which I don't think nobody gonna be on that because the mRNA is where it's at. So when we get this, we get the executive summary. We can break down the executive summary and fully understand what's going on here. Now, we might get something like this wrong because I don't expect all of us to start getting into that. You know, we look that up and we come up with, look, 1/1,000,000th of a gram. All right. 1/1,000,000th of a gram. Now, you'll see this symbol again, according to conventional vaccines, about how much, you know, a microgram is. And you know, you got people tripping on how much of, you know, a strand of the conventional vaccine had, I mean, how much of the, you know, that little bitty microgram, man, it's small stuff. And then they'll be like, man, it's got a microgram in it. Come on, man, just get out of here. But a strand of the virus in it. You ain't gonna shoot me up with that. I don't want no part of that. What's the one thing that people always used to cry about that you can get out of tuna? I don't know if you paying attention to take it out, but he said S protein is T-bone steak. Yeah, boy. But y'all know what I'm talking about, man. It's slipping my mind right now. But it's in breast milk too.
What you talking about?
What's in tuna that people always complain about? It has a high amount of what in it?
What's in what?
Tuna, mercury. There you go. Mercury. Yeah, that shit was slipping my mind like a mother. But anyway, you have mercury and tuna. You know, the earth got some form of mercury in it. You got some organic mercury. You got a different type of a mercury that can be traced in breast milk, right? And people complain about mercury showing up in a particular vaccine, not all vaccines, a particular vaccine. And they used to complain about that. Man, it's got high doses of mercury. Man, and that's causing autism and all of that. Remember, we was having those whole conversations. What people weren't understanding is the usage, like how much of this so-called organic mercury was in it. And it's just retarded. But as I was saying earlier, we began to start understanding things. There's one of those words again. Like the more that we actually get into what their lingo is, we can begin to redefine what some of these terms are. We can bring it down into street lingo, right? Our whole goal is to get this on a level to where it's feasible for us to communicate it, because they're gonna stay in a big word lane. They're gonna stay in a big word lane. But as long as we can pull out what we need to pull out for us to further understand what's going on, that's what matters the most. What you're looking at again for the listening audience and people are not familiar with Science with Shawn is the Moderna trial. Now, this document came out, was released to the public today. Some kind of weird way. We've been having this document for 2 days. You know what I'm saying? So we've been having it for 2 days. Now, these are things that are also important. So I am going to click here. Look at that. That's 18 pages for you right there, ladies and gentlemen. Then I'm going to bag out of back. And I'm going to get into this right here. And I'm going to scroll back down and I'm going to give you the other highlighted blue because that's important too. The license. That's 24 pages. You go all the way down, it's going to give you some other stuff that's going to be related to that. Now, remember, we've been having this document 48 hours before the release. So we ain't stupid. We're going to do the same thing with AstraZeneca. We're going to do the same thing with Johnson & Johnson. We're going to keep on trying to pull that homework out on everybody. And then we're going to deal with it from like that. But hold on. This guidance is being issued to address the coronavirus disease 2019 public health emergency. This guidance is being implemented without prior public comment because the FDA has determined that prior public participation for this guidance is not feasible or appropriate of the Federal Food, Drug, and Cosmetic Act. There go your act number. This guidance, the document is being implemented immediately, but it remains subject to comment in accordance with the agency's good guidance practices. You got questions, you email them, call them. In this one document, you got the intro, the background, the chemistry, manufacturing, control, and key considerations, non-clinical data, key considerations, clinical trials, key considerations, post-license safety evaluation, key considerations, diagnostic and serological assays, key considerations, and additional considerations. So everything is about considerations. You got efficacy considerations, statistical considerations, and so forth. So that's in this one right here. But we got the main course in the description. Right? This is the main course for Moderna. All of this is related to Moderna. The 2 links I put in the chat are Moderna driven. All right. So we need to know how they operate and how they move. Now, this was October 20th when this document was released. And this lets us know how they were moving toward trying to get the EAU. And this is the data surrounding that move. This don't have anything to do with the phase 3 trials. We got that, but this is further information. So I just want people to understand how they operate. They're being transparent. What other reason or way we would get this 48 hours in advance? And the last time we met, we talked about this.
On whether the benefits of Moderna's COVID-19 vaccine outweigh its risks to support authorization of the vaccine and an EUA for prevention of COVID-19 in individuals 18 years of age and older. So I look forward to the discussions and thank you.
At FDA, who will give us a presentation on emergency use authorization. Dr. Fink.
Hi, good morning. If the AC staff could please make me a presenter, then I will begin my presentation. In the meantime, I'll introduce myself. I'm Doran Fink. I'm the Deputy Director for Clinical Review in the Division of Vaccines and Related Products Applications, Office of Vaccines Research and Review, Center for Biologics Evaluation and Research at FDA. The COVID-19 pandemic continues to worsen in the US and worldwide. As of the week ending December 15th, there have been a total of 16 million cases and greater than 300,000 deaths in the US to date, and 1.5 million cases and greater than 17,000 deaths just in the past week. On December 11th, just last week, FDA issued an emergency use authorization for the Pfizer-BioNTech COVID-19 vaccine. This vaccine is authorized for active immunization for prevention of COVID-19 due to SARS-CoV-2 in individuals 16 years of age and older. The EUA was issued after a December 10th VRBPAC meeting to discuss the vaccine, data informing its benefits and risks, and plans for its further evaluation. On November 30th, Moderna Therapeutics submitted an EUA request for the Moderna COVID-19 vaccine, otherwise known as mRNA 1273. This, like the Pfizer vaccine, is an mRNA lipid nanoparticle vaccine, and it is administered as a 2-dose regimen 28 days apart. The requested use for this EUA is for active immunization to prevent COVID-19 caused by SARS-CoV-2 in individuals 18 years of age and older. The information submitted with the request includes safety and efficacy data from a large, randomized, blinded, placebo-controlled phase 3 trial. FDA has been conducting a comprehensive review of the Moderna COVID-19 vaccine EUA submission received on November 30th. As with the Pfizer request, our review has been comprehensive and conducted over a short period of time. We have verified clinical data integrity and integrity of Moderna's analyses and conducted our own independent analyses from data sets provided in the submission. We've conducted ongoing review of manufacturing, non-clinical, and clinical assay information, including information that has come in the last few days. We have reviewed and worked on revisions of prescribing information and fact sheets necessary to inform vaccine recipients and healthcare providers. We've had multiple information requests to Moderna to address our questions and need for clarifications. And of course, we have prepared for today's VRBPAC meeting. This will sound like a bit of a broken record, but I say it again because it's important. Today's VRBPAC meeting continues FDA's commitment to an expedited review process that is transparent, scientifically sound, and data-driven. As a reminder, from material presented last week, the legal authority for emergency use authorization was established in Section 564 of the Federal Food, Drug, and Cosmetic Act and allows for FDA authorization of unapproved medical products or unapproved uses of approved medical products to address public health emergencies related to biological, chemical, radiological, or nuclear agents. HHS Secretary Azar issued a declaration on March 27th justifying emergency use authorization of drugs and biological products to address the COVID-19 pandemic, which is a necessary prerequisite for issuance of an EUA. Here again are the criteria for FDA issuance of an EUA. The agent referred to in the EUA declaration must cause a serious or life-threatening disease or condition. Again, we know this to be true for COVID-19. The medical product must be effective or must be believed to be effective to prevent, diagnose, or treat the serious or life-threatening disease or condition caused by the agent. The known and potential benefits of the product should outweigh the known and potential risks of the product. And also there must not be any adequate, approved, and available alternative to the product for diagnosing, preventing, or treating the disease or condition. As I explained last week, there is only one FDA-approved product for COVID-19, which is remdesivir, approved for treatment and not for prevention. As I mentioned at the beginning of my talk, the Pfizer-BioNTech COVID-19 vaccine is now available under EUA for prevention of COVID-19. But it remains unapproved, and its quantity is not sufficient for mass vaccination needed to address the pandemic in the US. Therefore, the fourth criterion is still met. FDA explained in guidance and in the VRBPAC meeting on October 22nd our expectations for data and other information to support issuance of an emergency use authorization for a COVID-19 vaccine. This information includes data to demonstrate manufacturing quality and consistency. And similar to the case with the Pfizer vaccine last week, FDA has reviewed the manufacturing information provided by Moderna and found it to be adequate to support issuance of an EUA. We expect clear and compelling safety and efficacy data to support a favorable benefit risk of the vaccine when rapidly deployed for administration to millions of individuals, including healthy people. And finally, we expect plans for further evaluation of vaccine safety and effectiveness, as we did for the interim analysis, but we don't think that this should hinder in any way our confidence in the data.
And he'll be telling us about the considerations for placebo-controlled trial design if an unlicensed vaccine becomes available. Dr. Goodman is Associate Dean of Clinical and Translation.
I skipped ahead for a reason. I want y'all to kind of see what's going on with them, what's going on for 8. And remember, this stream is 8 hours long, and this all FDA stuff. and all you're gonna get is presentations. The whole 8 hours, you're gonna get some conversation, you're gonna get some grid graphs. You know what I'm saying? You get an intermediate period where they take a break, they come right back and they put more data in your face. This thing is classroom exhaustive. This is something that we don't have the patience to do because we want to microwave everything and we don't want to sit through stuff like that. Right? So this dude he's talking about considerations for placebo-controlled trial design if an unlicensed vaccine becomes available. So they're looking at every measure to try to resolve this pandemic as quickly and as fast as they can. Look, this dude is from the university, Stanford University, some of the top schools.
Research at Stanford University School of Medicine. Dr. Goodman.
Morning. Can you hear me fine?
Yes.
Okay. Terrific. So thank you so much for inviting me, and in fact, particularly for inviting me back again. I think the reason for that was presaged by the questions and talk just given, which is that the issues we'll be considering here are not just relevant for this vaccine, but for many trials currently ongoing and those in the future. So this arguably has potentially more long-lasting and effect than even the EUA today. I don't want to scare you with this title, which looks exactly the same as last week. I'm in fact not going to be giving exactly the same talk. I'll be picking up from where we left off last week and taking a bit of a deeper dive to give you more material for your discussion. And this is the outline. I'll very briefly remind you where we left off after last week, then go into the Moderna consent and proposal. Then we'll take a bit of a deeper dive into the deferred vaccination design, which we talked about last week. Then I'll discuss the evidential and ethical effects of both partial and complete unblinding of the placebo group, which are both under consideration right now. And then I'll have a final single slide on the evolution of design, which is a perspective I hope you will consider in your comments. And I already heard a suggestion of this from Dr. Gans in her last question. So let's go just to very briefly summarize where we were after last week. We had an ethical summary where we talked about the issue of ethical dilemmas as being a choice between two right actions or justified in different ways, which is certainly what we're going to be facing today, even more starkly. The importance of trust in the whole vaccine development process and prioritization, which enables us to do these clinical trials and could be withdrawn at any time. Talked a bit about the issue of context, where the ethical calculus depends not only on what we know and what we don't, but the availability of vaccine. And we talked about ethically relevant benefit in the sense that whether the placebo group was taking on such a risk, the actual proposal, which we will contrast here. So first of all, an observation is made that there's going to be a large number of folks who are eligible for early vaccine administration. This, in particular, 25% of the enrollees are healthcare workers. But here's the proposal. They will proactively reconsent participants who receive placebo and then offer them the vaccination. And then they will be observed unblinded for the rest of the entire 2 years. And adverse events will be captured. But basically they are proposing to simply unblind and immunize the placebo group. This is importantly different from last week's proposal, which was to wait until there was until they were eligible for receipt otherwise, and then if they asked, they would then be unblinded and immunized, and that all participants would be encouraged to stay in the randomized trial as long as possible and that everybody would be immunized at the end of 6 months. So this is different in multiple ways. First, that it's done immediately. Second of all, that they be unblinded, that actually is common to both, but that we don't wait for eligibility outside the trial. So this is a very, very, very important difference, and something that will have, I will argue, consequences not just for this trial, but for other trials of other vaccines. So,
all right, that nigga boring too, but I can listen to him. So here we go right here, real quick, y'all. This is important, right? This is Tal Zaks, the MD, PhD, Chief Medical Officer, Moderna in Texas. Remember, we talked about Moderna. We talked about their location in Dallas. Y'all remember when I brought that up? So here we have the Moderna guy on here, which is important. I think he's gonna give us a little life, but he's gonna give us something to, there you go. He's gonna give us a little life, and we're gonna get it to it.
Sure, go ahead. Thank you. Good morning. My name is Tal Zaks, and I'm the Chief Medical Officer at Moderna. On behalf of myself and my colleagues, I'd like to thank the committee and FDA for the opportunity to present our data today. We've carefully watched and listened to the meeting last week, and in preparing our presentation for today, we've attempted to proactively address many of the topics raised at that meeting and in the days that have followed. We have been developing our mRNA 1273 vaccine with a goal to seek global licensure for the prevention of COVID-19 disease, and are here today seeking emergency use authorization based on phase 3 safety and efficacy data. I don't need to belabor the damage this virus continues to wreak directly on our health and indirectly on our society and our way of life. Since the pandemic began, we at Moderna have moved rapidly to leverage the advantages of our mRNA platform, and we've been working closely with colleagues from the National Institutes of Health to develop our vaccine. We've done so in a very transparent manner, sharing our phase 3 clinical trial protocol as well as recruitment metrics with the public. Let me briefly explain the merits of our vaccine. mRNA 1273 is based on messenger RNA, a molecule that is fundamental to the biology of every living cell and serves as the blueprint for all protein synthesis. Our vaccine uses our body's own cells to activate the immune system. It enables these cells to make only the part of the virus that is critical for the immune system to recognize, in this case, the spike protein. Importantly, our vaccine platform has some inherent safety features. The mRNA does not self-replicate, does not enter the nucleus, and does not integrate into our DNA. The manufacturing process is cell-free. It does not use products of animal or human origin, and it does not contain preservatives or adjuvants, thus avoiding some of the potential concerns of older vaccine technologies. Now, mRNA 1273 is not our first infectious disease vaccine. In fact, we've been in early phase clinical trials for the past 5 years, conducting 12 clinical trials that have enrolled over 1,700 healthy.
Let me pause this real quick. Now, one of the reasons that I actually did this is because of this information right here. See, like I said, not many black people are going to watch 8 hours of this. They would have been clicked off this a long time ago, right? But today's Moderna's day. Just like last week during the FDA trials, it was Pfizer's day. And here we have the guy, the MD, the PhD, right there. Moderna got assistance, but they told you off the rip. We've been dealing with certain information like this. We had a collaborative effort for this particular vaccine with the NIH. If you don't know who the NIH is, you got to deal with Fauci. If you know who Fauci is, Fauci and them and the NIH now, because that's the head, he the head of the NIH. They got together with Moderna to create this vaccine, right? So he's telling you right now what's going on and what they've been doing for quite some time. Clinical experience with mRNA infectious disease vaccines since 2015. It is 2020, about to be 2021. Where's Unc at? I'm dropping these jewels on y'all, man.
healthy volunteers. SARS-CoV-2 is the 9th virus against which our mRNA vaccines have elicited neutralizing antibodies, and we have not seen a significant safety concern in any of our trials to date. Since the company's inception, we've been investing heavily in understanding the critical quality attributes of our mRNA medicines, and we have been using these insights to continuously improve our process and manufacturing capabilities. We've leveraged this progress at the start of the pandemic to develop a product that remains potent and stable in cold chain shipping and storage conditions that are widely available in hospitals, pharmacies, and assisted living and skilled nursing facilities. At the point of care, mRNA 1273 can be deployed in a multi-use vial with no further mixing or dilution, while remaining stable for up to 12 hours at room temperature. Our phase 3 study, which is the basis of our presentation today, was conducted in collaboration with the NIH and in accordance with clear FDA guidance. It enrolled over 30,000 participants, and we believe the results support emergency use authorization. mRNA 1273 efficacy clearly exceeds the recommendations for an EUA and eventual licensure. The vaccine efficacy rate for symptomatic COVID-19 infection was 94.1%, with a 95% confidence interval lower bound of 89.3%. These results are clinically meaningful and highly statistically significant. The efficacy observed is broadly consistent across all evaluated subgroups. Importantly, we also observed a dramatic reduction in severe cases. All of the 30 severe cases observed at the time of primary analysis occurred in people given placebo. A reduction in total symptomatic cases predicts a reduction in cases leading to hospitalization, intensive care, and death. Finally, data from 9 weeks of median exposure in more than 15,000 people vaccinated with mRNA 1273 have well characterized the short-term safety profile. We see generally good tolerability. Most solicited injection site reactions and systemic adverse events were reported as mild to moderate and resolved quickly. It is important to note and to educate people that we see an increased rate and severity of expected systemic symptoms like headache and myalgia after the second dose. We view these as consistent with a potent activation of a specific immune response. They are transient and self-limited, and we do not see a significant safety risk. These results support acceptable benefit risk for broad population vaccination to help prevent COVID-19 infections. We acknowledge the need for longer-term safety and effectiveness data. We will continue to transparently share our data, and the independent DSMB will continue to monitor safety as well as monitoring the duration of immunity and effectiveness. And we will continue to leverage the phase 3 trial even as we amend it to enable access to participants who received placebo. Now, in this regard, we face some unique circumstances. First, as it relates to vaccine supplies, none of our trial participants would be quote-unquote jumping the line ahead of others because we have clinical trial supplies available that in fact would expire and go to waste if we don't use them. Second, all of our participants are at increased risk of infection, and many have risk factors for severe disease. One of the participants on our placebo arm died from COVID-19 during this trial. He was a 54-year-old male whose sole risk factor was diabetes. I'll defer to Dr. Baden to describe our proposed next steps on the trial, which will continue to be overseen by the DSMB and should provide significant additional data on both safety and effectiveness. Now, beyond the phase 3 trial, Moderna will conduct additional studies in active pharmacovigilance to gain a more comprehensive understanding of the vaccine risk profile over time. We are initiating pediatric clinical trials, collaborating with the National Cancer Institute to evaluate vaccine safety and immunogenicity in people with cancer, and will continue to collaborate with FDA and other agencies to gather additional long-term safety data. Here now is the agenda for the rest of our presentation. Let me now turn it over to Dr. Melissa Bourne.
All right. So we heard his introduction. The key points was made at the beginning. They've been dealing with mRNA vaccines since 2015. So they've been working in the field on an mRNA vaccine for 6 years. But the mRNA science and technology and perspective of the mRNA been around for 25 years. That we know. So we all over it over here. I don't know what other channels teaching and preaching and all that other stuff. I ain't even studying that. But over here on Science with Shawn, we're going to get it in.
Hello, good morning. My name is Melissa Moore, and I am the Chief Scientific Officer of Platform Research at Moderna. I'm also a professor in the RNA Therapeutics Institute at the University of Massachusetts Medical School. Over the next few minutes, I will walk you through a description of Moderna's vaccine platform and specifically our COVID-19 vaccine, mRNA 1273. As the basis of our vaccine, we created a messenger RNA, or mRNA, that only contains the instructions to make the SARS-CoV-2 spike protein.
Did y'all hear what she said? Let me run it back, because she's teaching right now. I don't care what she look like, she's teaching right now.
Over the next few minutes, I will walk you through a description of Moderna's vaccine platform and specifically our COVID-19 vaccine, mRNA 1273. As the basis of our vaccine, we created a messenger RNA, or mRNA, that only contains the instructions to make the SARS-CoV-2 spike protein in a pre-fusion conformation. We manufacture this mRNA in large quantities in a cell-free process that utilizes no ingredients of human or animal origin. We then formulate this mRNA with lipids to form lipid nanoparticles, or LNPs. As can be seen in the electron micrograph at the bottom right, our GMP manufacturing process yields a highly consistent product, about 100 nanometers in diameter. In addition to the mRNA and lipids, the only other ingredients in the vial are water, sucrose, and 2 FDA-approved pharmaceutical buffers. Importantly, our vaccine contains no preservatives, no antibiotics, no adjuvants, and all components are rapidly cleared from the body. When our vaccine is injected intramuscularly, it is primarily taken up in the draining lymph nodes by specialized immune cells known as antigen-presenting cells, or APCs. Once inside the antigen-presenting cell, RNA instructs the cell's protein synthesis machinery to make the spike protein, which is then displayed on the cell surface. In the lymph node, this allows B cells and T cells to interact with the spike protein and develop an adaptive immune response. This adaptive immune response includes production of antibodies and the development of T cell responses against the spike protein, resulting in both humoral and cell-mediated immune memory. Once the mRNA has done its job, it is degraded. Importantly, our mRNA vaccine has no capacity to alter DNA. First, our externally delivered mRNA constitutes only a tiny fraction of all mRNA molecules in the cell. Second, our mRNA is transient and remains in the cytoplasm until eliminated by the natural mRNA decay process. To alter DNA, our mRNA would have to both gain access to the nucleus and be reverse transcribed. Our mRNA contains no signals for nuclear access and no known signals for reverse transcription. So, in summary, mRNA 1273 directly educates the immune system by instructing antigen-presenting cells to synthesize the SARS-CoV-2 spike protein. In this way, it efficiently drives an adaptive immune response by protein expression in situ. Finally, our mRNA can neither interact nor can it integrate into DNA. Thank you. I'll now pause and hand the presentation over to Dr. Jacqueline Miller to discuss mRNA.
Now, before we move any further, is that do I need to rewind this for anybody in the chat so they can properly understand what this lady just said in like 2 or 3 minutes? I know there's a delay, but I want to make sure that our people are properly informed about the messenger mRNA. That's all we've been talking about is the messenger RNA vaccine. And we led the show with that. And now we're listening to Moderna, their scientists, their doctors, their leads, educate us on this stuff from their perspective, telling you how this works. Her last slide is how this works. Provides instruction directly to the immune system, the spike protein. Efficiently creates specific immune memory in a natural context. The second bullet point is what we want. Efficiently creates a specific immune memory in a natural. Where are y'all holistic people at? This is creating a natural immune response. Chris said he got to Google some words, she said. mRNA can neither interact with nor integrate into your DNA. Man, look, I'm gonna run it back, y'all. I'm gonna run it back because I really don't think that y'all actually catching what she throwing.
Over the next few minutes, I will walk you through a description of Moderna's vaccine platform and specifically our COVID-19 vaccine mRNA 1273. As the basis of our vaccine, we created a messenger RNA, or mRNA, that only contains the instructions to make the SARS-CoV-2 spike protein in a pre-fusion conformation. We manufacture this mRNA in large quantities in a cell-free process that utilizes no ingredients of human or animal origin. We then formulate this mRNA with lipids to form lipid nanoparticles, or LNPs. As can be seen in the electron micrograph at the bottom right, our GMP manufacturing process yields a highly consistent product about 100 nanometers in diameter. In addition to the mRNA and lipids, the only other ingredients in the vial are water, sucrose, and 2 FDA-approved pharmaceutical buffers. Importantly, our vaccine contains no preservatives, no antibiotics, no adjuvants, and all components are rapidly cleared from the...
No preservatives, no antibiotics, no adjuvants, all components rapidly cleared. Because I know we got to hit that 4 or 5 times. Water, sucrose, FDA approved buffers. mRNA 1273 plus. Water, sucrose, FDA approved buffers. So I guess y'all gonna want to know, what are the FDA approved buffers?
The body. When our vaccine is injected intramuscularly, it is primarily taken up in the draining lymph nodes by specialized immune cells known as antigen-presenting cells, or APCs. Once inside the antigen-presenting cell, RNA instructs the cell's protein synthesis machinery to make the spike protein, which is then displayed on the cell surface. In the lymph node, this allows B cells and T cells to interact with the spike protein and develop an adaptive immune response. This adaptive immune response includes production of antibodies and the development of T cell responses against the spike protein, resulting in both humoral and cell-mediated immune memory. Once the mRNA has done its job, it is degraded. Importantly, our mRNA vaccine has no capacity to alter DNA. First, our externally delivered mRNA constitutes only a tiny fraction of all mRNA molecules in the cell. Second, our mRNA is transient and remains in the cytoplasm until eliminated by the natural mRNA decay process. To alter DNA, our mRNA would have to both gain access to the nucleus and be reverse transcribed. Our mRNA contains no signals for nuclear access and no known signals for reverse transcription. So, in summary, mRNA 1273 directly educates the immune system by instructing antigen-presenting cells to synthesize the SARS-CoV-2 spike protein. In this way, it efficiently drives an adaptive immune response by protein expression in situ. Finally, our mRNA can neither interact nor can it integrate into DNA. Thank you. I'll now pause and hand the presentation over to Dr. Jacqueline Miller to discuss mRNA 1273 efficacy.
Morning. My name is Dr. Jacqueline Miller, and I am the Senior Vice President and Therapeutic Area Head of Infectious Diseases at Moderna. I'm pleased to share with you today some of the details of our clinical development program and our key immunogenicity and efficacy results. Before moving to our clinical program, I would like to review our key non-clinical results. Generated an extensive non-clinical data package in 3 different animal models, including non-human primates, or NHPs. Our data demonstrate that mRNA 1273 induces humoral and cellular immunity, including memory B cells in vaccinated animals. We also challenged these animals SARS-CoV-2 virus and found that the vaccine fully protect animals at subtherapeutic doses. No evidence of vaccine-associated enhanced disease. We have recently completed our developmental and reproductive toxicology study, indicated no safety concerns. Development of mRNA 1273 has been accelerated given the COVID-19 pandemic. Nonetheless, a full development program, including phase 1, 2, and 3 studies, has been executed. Study 101 was our phase 1 dose-ranging safety and immunogenicity study conducted across 3 age strata, 18 to 55, 56 to 70, and over 71 years of age. Study 201 was a phase 2 safety and immunogenicity study. The primary focus of our presentation will be the phase 3 COVID-19 efficacy and safety study, or 301, as it enrolled over 30,000 participants, approximately 15,000 of whom received mRNA 1273. Study 301 generated the vast majority of safety and all of the efficacy. So let's begin with Study 101. This slide summarizes the neutralizing antibodies induced by 100 micrograms of mRNA 1273 across 3 age strata. Shaded area represents a panel of titers, a range of titers from a panel of convalescent sera taken from individuals recovering from COVID-19 disease. Served as the clinical benchmark to compare immunogenicity between the doses and the samples were 23 to 54 days after diagnosis. Neutralizing antibodies were induced in all participants by day 36, or 1 week, dose 2. GMTs were comparable across the 3 age strata, including participants in the older age strata, and persisted until day 119. Now, let's discuss the T-cell immunity evaluated in Study 101. CD4 T cells were further evaluated for Th1 and Th2 phenotypes, since T cells are thought to be associated with enhanced disease. Top panel of this slide represents the Th1 phenotype, and the bottom panel is the Th2. Th1 dominant CD4 T cells are induced by day 43 across age strata. Minimal detection of the Th2 phenotype. This analysis showed no evidence of enhanced. I'd now like to present the immunogenicity results for the 100 microgram and placebo groups in the Study 201. Dark blue bars represent the 100 microgram dose, and the gray bars represent placebo. By day 43, there was more than a 50-fold increase in geometric mean titers in the vaccine group. And in the placebo group, GMTs remained below the level of quantitation. So in summary, our phase 1 and 2 studies showed the induction of neutralizing antibody titers in all participants by 1 week following the second dose. GMTs were observed to be higher than those of a panel of convalescent sera, and neutralizing antibodies persisted for 3 months after the second dose across all 3 age strata. Th1 dominant CD4 T cell response was also observed across age strata was consistent with our findings in animal models. So now, let's look at the efficacy data from Study 301. Study 301 was designed to evaluate the efficacy, safety, and immunogenicity of mRNA 1273 compared to placebo in adults at least 18 years of age who were at risk for COVID-19. 30,420 participants were randomized 1-to-1 to receive 2 doses, vaccine or placebo. Participants received the first dose on day 1 and the second dose 1 month later on day 29. Participants have been monitored for efficacy, immunogenicity, and safety endpoints throughout the study. Immunogenicity endpoints include the measure of binding and neutralizing antibodies at the indicated time point. These immunogenicity samples will also be used to assess for asymptomatic seroconversion non-vaccine antigen. These data were not available for the emergency use submission and will not be discussed for today. Efficacy surveillance occurred throughout the study. Once diagnosed with COVID-19, participants underwent daily telemedicine visits to ensure close medical follow-up. Participants were also given pulse oximeters to measure their oxygen saturation daily. Study 301 primary efficacy objective was based on COVID-19 cases that occurred in SARS-CoV-2 seronegative participants. Demonstrate success, the lower limit of the 95% confidence interval for vaccine efficacy had to be greater than 30%. Secondary endpoints for vaccine efficacy include the evaluation of efficacy against severe disease and death, COVID-19 using the CDC case definition, and COVID-19 cases occurring after the first dose. There was also a secondary objective to evaluate asymptomatic SARS-CoV-2 infection, but the results are not yet available. Please let me review the case definition for COVID-19, severe COVID-19 disease. Primary efficacy endpoint was symptomatic adjudicated COVID-19 disease that occurred at least 14 days after dose 2. To be considered a case of COVID-19, study participant had to have experienced at least 2 systemic symptoms or at least 1 respiratory sign or symptom or clinical or radiographical evidence of pneumonia and confirmed SARS-CoV-2 infection from at least 1 nasopharyngeal. Study 301 also analyzed efficacy against severe COVID-19. Severe cases had to meet all criteria for the primary endpoint and have at least one of the 4 following criteria: severe systemic illness or respiratory failure, acute respiratory distress syndrome or evidence of shock, or significant acute organ dysfunction or admission to an ICU or death. To ensure adequate safety monitoring and to enable the interim efficacy analyses, this study has been monitored by a Data and Safety Monitoring Board, or DSMB. DSMB was chartered and convened by the National Institutes of Health and is completely independent from the company. In addition, an independent efficacy endpoint adjudication committee was assembled to determine if the case definitions for COVID-19 and severe COVID-19 were met. The committee has adjudicated all cases for the primary efficacy endpoint and continues to adjudicate cases as they accrue and will ultimately adjudicate all COVID-19 cases reported. 30,420 participants were randomized in Study 301, including 15,010 subjects to each group. The full analysis set includes 15,181 participants who received at least 1 dose of mRNA 1273. The modified intent to treat population includes participants who had no evidence of infection prior to receiving their first dose of study vaccine or placebo. For protocol population, predefined for the primary efficacy analysis. It includes participants in the MITT who received both planned doses and had no major protocol deviations. More than 92% of participants vaccinated in both treatment groups are part of this population. Now let's return to the efficacy results. Enrollment was stratified to ensure that we studied participants most at risk for COVID-19. We pre-specified that at least 25% of our study population would include participants over 65 years of age or subjects between 18 and 65 with comorbid medical conditions. We were successful and enrolled a total of 42% of the study population in these two categories. Let's review the study demography by gender and age. Approximately equal proportions of males and females participated, and the mean age was 51, the range of 18 to 95 years. 25% of the study population was over 65 years of age, and half of those individuals were over 70. Age and gender distribution were well balanced. This trial included approximately 10% African Americans, 5% Asian American, and 21% of participants who identified as being Hispanic. This is a breakdown of the comorbid conditions reported in the study. 23% of participants overall reported at least one pre-existing condition. This included 9% with diabetes mellitus, 7% with severe obesity, 5% each with significant cardiac disease or chronic lung disease. A specific inclusion criterion was that participants had to be at increased risk for COVID-19. Overall, 25% of our study participants are healthcare providers, and a substantial proportion of the remaining subjects meet the definition for essential workers. Taken together, the participants depicted on this table represent more than 50% of our study population. So here are the numbers of COVID-19 cases contributing to the primary endpoint by demographic subgroups. 33 cases occurred in the elderly, including 10 of the severe cases. 42 cases occurred in people from communities.
We'll pause there real quick. Her internet whack is on over because she breaking up left and right. However, she goes on through, and then after she get done, it's another dude up. This whole thing is about Moderna, and then they take a break. I'm not going to bore y'all with all of that because it's also in the link, and y'all can catch up to that. I see we're having a good time in the chat, and that's what it's all about. But hopefully when it comes down to the conversation, we actually can do what we need to do in order to combat a lot of the misinformation. I do see that Robert Rand said in the chat that the ABOs had a solution to something. And what solution do they have? Because if they got a solution, they need to write the paper and submit it. And here's where you can handle all your business. Right here. For written paper comments submitted to the documents manager, look, they telling you what to do. Instructions. All submission received must include the docket number, FDA 2020. All that, there you go. Vaccine, all that, put all that in there. All that in there. All of that in there. They running around. Got a whole lot to say. Just tell them submit their work. Submit their findings to the FDA. Submit it to the scientific field. Let's see who playing the game real, raw, and all that. Come on, man. We ain't got time, man. They presenting facts over here, man, and we wasting our time with eyeballs, yo. They ain't trying to do nothing. Like I said, all sourced information in this particular build is in the description. I done ran Sutek up out of here. I'm here by myself. I appreciate everybody tuning in and rocking out with me, man. Look for Science with Shawn tomorrow. We're going to get into a continuation of a build. We might do it tomorrow. You never know. But I appreciate everybody's time and energy. books is in. The books are in. Yep. They in. Mm-hmm. They're here.